Hello guys, welcome back to the channel Biopulse, and this is Chandrika. Today we are on the journey of day 35, complete biology MCQ in 40 days. We are near to our goal, isn't it? And I'm very happy about that. And we are discussing about the chapter Biotechnology and its applications, one of the most interesting chapter I can say. It's not just about the understanding life at the molecular level, that is biotechnology. It's about using knowledge to solve the real world problems. In this chapter we explore how biotechnology is used in the fields like medicines, agri, and then industries to improve human life, from producing life saving medicals like drugs like insulin, to developing genetically modified crops with high yielding and pest resistant. This chapter focus on two major areas, that is applications in agriculture and in the medicines. If I talk about biotechnology applications in agri, GM crops, pest resistance, and if I'm talking about the medicines like recombinant proteins, vaccines, and gene therapy. So, for the sake of the NEET, you may expect 2 to 3 questions from this chapter. Focus mainly on genetically modified crops, that is GM crops, BT cotton and pest resistance, recombinant insulin, and gene therapy, vaccines, and molecular diagnosis. These areas are very specifically focused area in this chapter. So, let's move towards the discussion area. Here comes our first question, that is from the basis of classification. First one, biotechnology mainly deals with, let me take a pen. Biotechnology mainly deals with industrial scale production of biopharmaceutical. B, biological use of genetically modified microbes, fungi, plants, and then animals. Both A and B, none of this. So here the right option is option C. Both A and B are right. Next, second question, which of the following is not included in the application of biotechnology? A, waste treatment. B, conventional hybridization. C, energy production. D, genetically modified crops. So here, which of the following is not included, he is asking, application of biotech. So conventional hybridization is not included. Is a traditional method of plant and then animal breeding, so it is not applicable here. Next, third one, application like bioremediation, processed food, and therapeutics and diagnostics are related to biochemistry, microbiology, biotechnology, medical science. So they are related to biotechnology. Fourth question, dash is or are the critical research areas of the biotechnology? Creating optimal conditions for the catalyst functions, right? Providing best catalysts, this is also right. Developing downstreaming processing technique, this is also right. So the right option is D. Next, from biotechnology applicants in agriculture. Fifth question, which of the following is not for increasing food production? Not for increasing food production. A, agrochemical based agriculture. He is asking not for the increasing food production, this is used for that. Organic agriculture, genetic engineering, crop based agriculture, none of this. So they all are used for increasing food production, so none of this is right. Sixth question, organic agriculture is a technique of raising crops for increasing food production, this one itself is right. Seventh one, Use of genetically modified crops in crop field may reduce the harmful effects of the fertilizers, yes, maximize the yield, be environment friendly. All of this is right. Eighth one. Plants, bacteria, fungi, and animals whose genes have been altered by manipulation are called pest resistant organisms, hybrid organisms, genetically modified organisms, insect resistant organisms. They are called as what? Genetically modified organisms. Next, ninth question. Golden rice is genetically modified crop plant with incorporated gene meant for biosynthesis of vitamin E, K, omega 3, vitamin A. So here the right option is vitamin A. So golden rice is genetically engineered variety of the rice, that is Oryza sativa, designed to biosynthesize beta carotene, which is a precursor of vitamin A. Next, 10th one. Dash produced by Bacillus thuringiensis. T toxin, Bt toxin, and acid, all of this. The right option is B, Bt toxin. Bacillus thuringiensis. Bt stands for Bacillus thuringiensis. Next, 11th one. The bacterium Bacillus thuringiensis produce dash plants which reduces the amount of dash used. It's a insect resistant and insecticides are used. Insect resistance and insecticides are used. So D is right. Next one, 12th question. Which of the following crops are modified using Bacillus thuringiensis? Corn and cotton, right. Tomato and rice, of course. Potato and soybean, also right. So all of them are right. Which of the following is being grown in India by farmers as BT crops? Maize, brinjal, cotton, soybean. In India, specifically cotton. 14th one. By inserting a piece of DNA from dash, insect resistant transgenic cotton has been produced. A wild relative of cotton, bacterium, an insect, a virus. By inserting a piece of DNA from bacteria, insect resistant transgenic cotton has been produced, that is BT cotton is produced. Bacillus thuringiensis is a bacteria. 15th one. Some strains of Bacillus thuringiensis produce proteins that will insect like lepidopterans, coleopterans, dipterans, and all of this. So this is the right option is D. Next, 16th one. Coleopterans examples are flies, mosquitoes, beetles, all of this. It is examples for beetles. Coleopterans example is beetles. Flies and mosquitoes belongs to the dipters. 17th one. Bacillus thuringiensis forms protein crystals which contains a simple protein, non-toxic insecticidal protein, toxic insecticidal protein, simple lipids. So it is a toxic insecticidal protein. 18th one. Why does Bt toxin protein crystal not kill the bacillus? Because the bacteria encloses toxins in special sacs. Bacteria are resistant to the toxin. Toxin occurs as inactive protoxin in the bacteria. This is right. 19th one. Bt toxin kills insects by inhibiting protein synthesis, generating excessive heat, creating pores leading to cell swelling and lysing in the midgut of epithelial cells. This one is right. 20th question. The choice of genes of Bacillus thuringiensis incorporated in the crop depends upon A. Crop, yes. B. Targeting pest, yes. So both A and B are right. So 20th right option is C. 21. The crops having cry genes need small amount of fungicide, large amount of pesticide, small amount of insecticides, and then D, none of the above. So they won't need any kind of the fungicides, pesticides and insecticides. 22. The Bt toxin protein obstructs a biosynthetic pathway, causes death of insects, yes, stops eggs laying of adults, generating excessive heat. It causes death of the insects. 23rd one. Cotton bollworm controlled by Cry1AC, Cry2AB, Cry1AC, Cry2AC, Cry1AB, Cry2AC, Cry1AB, Cry1AB. Here the right option is option A, Cry1AC, Cry2AB. Cry1AC and Cry2AB, these are specific genes produced toxins that are effective against the cotton bollworm, whereas Cry1AB is for corn borer. Next, 24th one. Bt corn has been made resistant to corn borer by introducing of the gene, just now I said, that is Cry1AB. 25. Cry2AB and Cry1AB produce toxins that controls. This is Cry2AB for cotton bollworm and Cry1AB for corn borer. Cotton bollworm and corn borer respectively. A itself is right. Next question, question number 26. Which of the following nematodes infects the root of the tobacco plants, which reduces the production of tobacco? Melodogyne incognita, this one itself is right. Next question, 27th one. A novel strategy was adopted to prevent Melodogyne incognita infection in the tobacco plant that was based on the process of DNA interferons, RNA interferons, RNA initiation, DNA initiation. So here the right option is RNA interferons. That is called as what? RNAi. 28th one. The resistance against a nematode was introduced by implying RNA in tomato, Bt corn, Bt cotton, tobacco. Here the tobacco is right. 29th one. RNAi stands for RNA interferons, inactivation, initiation. So here interferons is right. The right option is B. RNAi stands for RNA interferons. 30th question. RNAi takes place in all organisms as a method of prokaryotes, insects resistance, no. B. Eukaryotes, insects resistance, no. C. Eukaryotes and cellular defense, yes. RNA takes place in all eukaryotic organisms as a method of cellular defense. It's a defense mechanism. 31. Dash is used for silencing of an unwanted gene. RNA is used for silencing of an unwanted gene in the RNAi process. 32. Silencing of mRNA molecule in order to control the production of an harmful protein has been used in the protection of plant from beetles, armyworms, budworms, nematodes. Here the right option is nematodes. 33. Transposons are known as, it is also known as silenced gene, Pleiotrophic gene, mobile genetic elements, both A and C. Here the right option is mobile genetic elements. 34th, the tobacco plant resistant to a nematode have been developed by a introduction of DNA, and it is produced in the lost cells as a particular hormone, B toxic protein, C both sense and antisense strand, D an antifidant. So here the right option is option C. 35th question from Biotechnology Applications in Medicines. 35. The first human hormone produced by the recombinant technology is estrogen, progesterone, thyroxine, insulin. Right option is option D, insulin. Next, 36. The demerits of using bovine insulin and porcine insulin in diabetic patients is it leads to calcemic, no, it may cause allergic reaction, it is expensive, all of this, it may cause allergic reaction. Right option is that one, because insulin for diabetic patients has extracted from the pancreas of cows, that is slaughtered cows, isn't it, and pigs also. Porcine insulin from pig and bovine insulin from cow. While this insulin was effective at the managing blood sugar level, it had several drawbacks, that is allergic reactions and animal sourcing, and today's modern solution we can use for them as that is RDNA technology we are going to use nowadays. 37th one, the two polypeptides of human insulin are linked together by phosphodiester bonds, no, disulfide bridge, yes, this one is the right. Next, 38th one, dash is removed during maturation of proinsulin to insulin. Which chain is removed? A chain, B chain, C chain, both A and B. No, it is only removed C chain. Next, 39th question. The main challenge for production of insulin using RDNA technology was option A, splitting A and B peptide chains, B, addition of C peptide to proinsulin, C, getting insulin assembled to mature form, removal of C peptide from the active insulin. So here the right option is option C. 40th question. Which of the following companies prepared human insulin in 1983? B. A. Monsanto, B. Eli Lilly, C. Genentech, and then D. GEAC. So here in 1983 the American company Eli Lilly becomes the first to produce and then marketed genetically engineered human insulin, which is called Humulin. Next one from the questions from gene therapy. 41st question. Treatment of genetic disorder by manipulating gene is called A. Gene therapy, B. RDNA technology, and then C. Bone marrow transplantation, D. Enzyme replacement therapy. So here the right option is option A, gene therapy. Whereas RDNA technology, we are that is called a recombinant DNA technology, is a tool used to gene therapy. It is a tool actually used for the gene therapy. Bone marrow transplantation, it is used to treat the genetic blood disorders, and whereas enzyme replacement therapy it is used to treat the symptoms by providing the missing enzyme rather than fixing the underlying genetic causes. So the right option will be option A. 42. For the first time, therapy was tried on a 4-year-old girl in 1990 to treat cystic fibrosis. No. Adenosine deaminase. Adenosine deaminase, that is ADA. In 1990, the first clinical gene therapy was performed on the 4-year girl to treat a deficiency of the enzyme adenosine deaminase. Okay, it is deficiency leads to like SCID, that is severe combined immunodeficiency, where it is a condition where actually the immune system is non-functional, making a person very highly vulnerable to infections. So this was the first to treat the gene therapy by. Next, 43. Which kind of therapy was given in 1990 to a 4-year-old girl with enzyme deficiency? That is, it is by a gene therapy, of course. 44. Adenosine deaminase, that is ADA deficiency, can be treated by dash and then dash, but it is not fully curated. Here A and B can be. A and B can be. A, that is bone marrow transplant, and then B, enzyme replacement therapy. Enzyme replacement therapy. So here the right option is option B. B is enzyme replacement therapy, and then A is bone marrow transplant. 45th question. The advantage of beginning gene therapy prior to birth is the body would not reject it as it has not yet recognized self. This would give the body plenty of time. The cell being extremely young are more receptive to the gene therapy. So here the right option is option A. Next, molecular diagnosis. 46th question. Why using conventional method for diagnosis is not very relevant? Early detection is not possible, not reliable, results are incorrect, all of this. So here the right option is option A. Early detection is not possible by conventional methods. So next, 47th one. Which of the following molecular diagnostic technique is used to detect the presence of a pathogen in its early stage of infection? Angiography, radiography, enzyme replacement technique, polymerase chain reaction. So here the right option is polymerase chain reaction. 48th. Why PCR is used? Why PCR is used? To detect HIV in suspected AIDS patients. Yes, it is also used here. To detect mutation in genes of the suspected cancer patients. It is also here. Diagnose many genetic disorders. Right. So all of the above is correct. 49th one. A single stranded nucleic acid tagged with a radioactive molecule is called plasmid, probe, vector selectable marker. Here the right option is option B, probe. 50th question. In which of the following method a probe is allowed hybridized to its complementary DNA in the clone of the cells? A, enzyme linked immunosorbent assay, that is ELISA. B, PCR. C, autoradiography. D, gene therapy. Here the right option is option C, autoradiography. It's a technique where a single stranded DNA or else RNA also you can take, tagged with the radioactive molecules, which is also called as probe. Next, 51. The technique used to detect mutation in genes is known as gel electrophoresis, no PCR, gene therapy, autoradiography. So here the right option is option D, autoradiography. 52. Which of the following technique is based on the principles of antigen antibody interaction? A. PCR, B. ELISA, C. Recombinant DNA technology, D. Gene therapy. Here the right option is option B, ELISA. Next, transgenic animals. 53 questions. Animals whose DNA is manipulated to possess and express an extra gene are known as transgenic animals. 54th one. Transgenic animals are those which have foreign DNA in all of their cells. Yes, this self itself is right. 59th question. 95% of all the existing transgenic animals are pigs, cows, mice, all of this. 95% of them are mice. 56. Transgenic animals can be used to study normal physiology. Yes. Study biological effects. Yes. Study the vaccine safety. Yes. So all of this is right. 57th one. Transgenic animals made to serve as models for human diseases. The diseases are A. Alzheimer's disease. Yes. For the cancer. Yes. For the cystic fibrosis. Yes. So the right option is all of this. 58th one. Which of the following transgenic human protein product development are used to treat emphysema? For emphysema, alpha 1 antitrypsin. First A itself is right. 59th one. When was the first transgenic cow, Rosie, produced? In 1979, 1997, 1996, 1999. So the right option is 1997. 60th one. Dash was introduced in the first transgenic cow. A. Alpha 1 antitrypsin. B. Human beta lactalbumin. Yes, this is used. Beta 1 antitrypsin, none of this. So the right option here it is B. The correct option is B, human beta lactalbumin. Next, 61st question. 61. The first transgenic cow, Rosie, produced human calcium enriched milk 2.4 gram per liter. No, human protein enriched milk. Human protein enriched milk 2.4 gram per liter. Human calcium enriched milk. Human protein enriched milk 2.6 gram per liter. 2.8 gram per. Here the right option is it produce human protein enriched milk 2.4 grams per liter. That is alpha lactalbumin. It's a protein. 62th question. Dash are used in testing safety of polio vaccine before they are used on a human. Transgenic pig, monkey, rabbit, mice. Here the right option is option D. 63th question. Dash animals are made that carry genes which makes them more sensitive to toxin substance than non-transgenic animals. A. Transgenic, mutants, transferals, and then transformed. So here the right option is transgenic. From the ethical issues, 64th question. Which committee takes decision regarding the validity of genetically modified research and the safety of the introducing GM organisms for the public service? A. Indian Council of Medical Research, that is ICMR? No. Genetic Engineering Approval Committee, that is GEAC. Indian Institute of Science Genetic Engineering Appraisal Committee. So here the right option is appraisal committee, not the approval committee. So GEAC. See, just now me also got confused, so don't get confused like that. Firstly, it was known as Genetically Engineering Approval Committee only, then it was changed to Appraisal Committee. So don't get confused among that. So option D is right here. 65. A dash granted to a person who has either invented a new and useful product, made improvement existing product, or invented a new process of the making a product is called bioethics, patent, biopiracy, genetic recombination. It is called what? Patent. It's a legal right or a monopoly, we can say, granted by a government to an inventor. 66th one. Biopatent means a right to use an invention, yes, a right to use application or processes, yes, both are right. So the right option is C. 67th one. Dash have been present in India from long time, yet foreign country got patent through the US Patent and Trademark Office. Brown rice, basmati rice, Co 667, all of this. So here this basmati rice is used been by foreign countries from the long time ago because of its fragrance. Next, 68th one. Bioethics. Bioethics is a process of discovery and commercialization of the new products, use of bioresources with proper authorization, standards used to regulate human activities in relation to a biological world. So the right option is option C. Next, 69th question. Exploitation of bioresources of a nation by multinational companies without authorization from the concerned country is referred to bioethics, bioweapons, biopiracy, bioexploitation. So it is with regard biopiracy. Next, 70th one. Biopiracy is related with the stealing of bioresources, yes. Traditional knowledge and then utilization, no. Biomolecules and regarding bioresource exploitation, this is not also right. So A and C is right. The right option is option D. So this is the last question for today's session. I hope you people are clear about the concepts with regarding biotechnology and its applications. So if you have any requests or else any comments, just drop your message in your comment box. Please do like, share and then subscribe my channel. Bye bye. Thank you one and all.